Welcome everyone to the History of Medicine Society lecture today. I'm Grover Bagby, the founding director of the Knight Cancer Institute here, and I'm here to introduce to you Jessica Wapner. A number of years ago, when I was division head in Hematology and Medical Oncology in the Department of Medicine, the president of OHSU, Peter Kohler, asked if I would be willing to try to convince the National Cancer Institute to bless us as an NCI designated cancer center. And I was glad to accept that challenge. It was a challenge, but I accepted it because I knew that we could be leaner and meaner and different. And what I mean by different is that in those days, the bulk of cancer research had to do with radiation, cytotoxic chemotherapies that were capable of killing cancer cells, but killed a lot of other cells too. And surgery, and those are the three major modalities, and they still are. They still are important modalities to do. And the way cancer research was done then is that we had phase 1, phase 2, phase 3 clinical trials of this poisonous thing and that poisonous thing, and if they didn't work, we started adding 2 together and then 3 together and then spacing them apart in a different way. These were called empirical clinical trials. The fundamental philosophy in those days was a scorched earth policy, and that is that if you have a cancer, you need to just kick the heck out of it and kill the cells. And if there's some collateral damage along the way, so be it. The vast majority of clinical trials in the 60s and 70s were failures. That is, the new idea, the new way, proved to be not any better than the old way. The successes were pretty meaningful in human terms. The Hodgkin's disease became incurable to curable with chemotherapy and radiation. Testicular cancer, from empirical trials, was converted from a fatal disease to a generally curable disease. And acute lymphocytic leukemia of childhood was better also. Now that took a long time because there were incremental advances, a few weeks more survival, a few weeks more survival with each trial. But these did work, but the majority were failures. I believe strongly, and there were some of us around the country that did, that there were better therapies that were possible, but they needed to be targeted in a way that required that we know exactly what causes the abnormal growth and invasion of cancer cells. And that a knowledge of exactly what was wrong at a molecular level was really, truly required for us to develop any kind of chance of going after those lesions. When we first asked the National Cancer Institute to visit our campus, we said, okay, here's the way our cancer center works. And I presented our operating principle, which was that we were going to focus on basic science and find out what was fundamentally wrong with each cancer, and then design things that were going to use that information to control the disease and cure it and put it into remission. The majority of the site visit team were members of what I called the old way of doing business club. And that is, they knew how to run empirical clinical trials and really did a lot of eye rolling when I presented this idea. And I knew what they were thinking. They were thinking, oh, here's another MD who has a lab and thinks that the lab is going to answer everything. Fortunately, I had a secret weapon, and that was a young guy from Harvard who was my first recruit. And by the name of Brian Drucker. Not only did he share a belief in this operating principle, that is fundamentally, if you understand it, you can fix it, but he was actually doing research that was apt to lead to a clinical trial of that kind of treatment. I don't need to tell you the rest of the story because I'm going to leave that to Jessica Wapner, our speaker today. Her book, The Philadelphia Chromosome, was ranked by the Wall Street Journal as in the top 10 of nonfiction works of 2013. And as I read this book, after I read it, I immediately bought 7 copies to send to my son, my daughter, my grandson, and my brother-in-law, and blah, 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 from here to the United Kingdom, friends in the United Kingdom. Because if you want to know about OHSU and our cancer center, this one right here, and why we're different, or if you want to know about the state of cancer research, BD and AD, by which I mean before Drucker and after Drucker, or if you want to know how exciting academic medicine can be, being on the ground. If you want to know what tensions can exist between the market wings of the pharmaceutical industry or the scientists of the pharmaceutical industry, and most importantly of all, if you want to know how it feels to be a patient or a parent of a patient with a fatal disease that might be involved in this kind of unique targeted clinical trial, you need to read this book. Honestly, it's the best book of this kind I've ever read, and I don't mean in the last year, I mean ever in my life. So I'm very, very happy and honored to introduce to you our speaker today, Jessica Wapner.
Hi, thank you so much. That was a really nice introduction. Thanks all for being here. Thank you, Maya Anderson. Oh, hi. Thank you so much, Maya Anderson and the History of Medicine Society for having me here. I'm really, really pleased to be back in Portland, which is where my research began for this story, which was in February, so yay, May. So I'm going to start with a book, reading an excerpt from the book, and then talk about why I wrote the book from there, okay? So I will begin with an excerpt. It's just from the Prelude, so for anyone who hasn't read it, I'm not giving too much away, okay? Okay, so it begins, Down to the Bone, February 2012. Gary Eichner sat in a chair backed up against a wall. Across the room, his nurse was half hidden by a computer. She scrolled through his medical chart on a monitor he couldn't see, typing his responses to her questions. He answered and smiled as if he were just fine, as though that would somehow make it so. He kept his worry concealed, silently wondering what the next hour would reveal about the disease that had so suddenly overtaken his life. Fear permeated his every thought. The nurse ran through the litany of side effects that he might be experiencing from the leukemia medication he'd recently begun to take. Any chest pain, she asked? Heart problems? Any swelling in your ankles? Any problems with nausea? Eichner's blanket no was tempered only by his description of what happened when he took the drug on an empty stomach. It's massive cramping, massive pain in the stomach, he told the nurse. It's just like the worst thing you've ever had. The nurse knew that Eichner really had no idea of the worst thing someone with his type of leukemia could have. Few patients with Eichner's disease today will ever know that kind of pain. But if the nurse was thinking anything of the sort, she kept it to herself. After all, her patient was about to have a large hollow needle inserted into his bone so that his doctor could extract a sample of his marrow. Having witnessed countless such procedures during the 12 years she'd been a nurse to patients taking the drug, she knew how much was riding on this biopsy, and she knew that Eichner, age 43, was well aware that his life was at stake. If anyone could set him at ease about that, it was his doctor, Brian Drucker. "All I care about is that you're making some progress," Drucker told his patient, who had come to Drucker's clinic in Portland, Oregon for a bone marrow biopsy. The procedure would reveal whether the medicine, a pill he'd been taking daily for 6 months, was tackling the leukemia that had invaded his body. Eichner had chronic myeloid leukemia, or CML, a cancer of the white blood cells that, though slow-growing, could be fatal. "I will be happy if you're 18 out of 20, Eichner nodded his understanding, his jittery foot the only sign of his nervousness. For Eichner, those numbers were part of the new language he'd learned since his diagnosis in the summer of 2011. As with so many cancer diagnoses, the education began suddenly and unexpectedly. After a day or so of sharp, excruciating kidney pain, Eichner, a single parent of a teenage boy, drove himself to the emergency room at his local hospital in Olympia, Washington, where he'd been living at the time. His sister-in-law, a trauma flight nurse, had told him she thought he had kidney stones, so Eichner was expecting to be admitted for a standard, albeit painful, procedure. But when several doctors walked into his hospital room together, he knew something wasn't right. "You don't have kidney stones," they told him. "You've got what we believe is leukemia." A blood test soon revealed that Eichner, in otherwise perfect health, had CML. The excess number of white blood cells contained in the sample confirmed that much. The doctors explained that the stabbing pain he'd thought came from his kidneys probably was from his spleen, enlarged by the high concentration of leukemia cells within. Although the slow-moving nature of the disease meant he wasn't in immediate danger, there wasn't any time to lose. For the best chance at long-term survival, he needed to start treatment right away. As Eichner was rapidly informed, danger could eventually come. If the treatments didn't work, within 5 years his bone marrow would fill with blast cells, white blood cells that fail to mature and thus are both overly abundant and thoroughly useless. His blood, once free-flowing, would turn into viscous sludge. The supply of iron-rich red blood cells that carry oxygen around the body would steadily plummet, leaving him fatigued and anemic, while a decrease in the number of platelets would render his blood unable to clot. As the disease progressed, the minuscule capillaries leading to his eyes and brain would clog. His spleen would likely become profoundly enlarged. As his body began shutting down, he would bleed in his brain, in his intestines, and out of every orifice. 2 days later, Eichner was having his first bone marrow biopsy, performed by a nurse who spoke broken English and had to climb on top of Eichner to hammer in the 4-inch-long needle to break through his bones, which had become hardened and inflamed from the profusion of white blood cells inside. Finally, she managed to extract an ounce of marrow, the spongy matter in the center of our bones where new blood cells are made. In the genetics laboratory, a fluorescent dye applied to the coiled strands of DNA inside Eichner's blood cells revealed the telltale sign of CML, a genetic mutation known as the Philadelphia Chromosome, an abnormal chromosome regarded as the defining feature of this fatal, spontaneously arising cancer, and discovered more than 50 years ago. 20 out of 20 cells in a sample of his marrow contained this genetic error. A family friend with the same type of cancer told Eichner to call a doctor named Brian Drucker right away. "Don't do anything before you see him," the friend told Eichner a couple of days after his diagnosis. 3 days later, Eichner got a call from Drucker, who'd mysteriously, Eichner didn't know exactly how, gotten his phone number. During the next 20 minutes, Drucker talked Eichner down from his panic, assuring him that, being in the early stages of the disease, Eichner could wait a week or two before making the trip south to Oregon Health and Science University, where Drucker had been treating and researching leukemia since 1993. He gave Eichner his professional blessing to go drink beer. It was August, and Eichner had been getting ready to leave for a day at Olympia's annual Summer Brew Fest, his brother's idea for getting his mind off leukemia for a while, when the phone had rung. 10 days later, Eichner was on his way to Portland. Within 3 weeks of his diagnosis, he was swallowing his first tablet of medication, which Drucker had been instrumental in developing, a drug aimed at tackling the cancer at its root. So I'll leave off there. I won't leave you in complete suspense. Gary Eichner is alive and well still. That test that he was having that day, which I had been here observing, came back with 0 out of 20 cells positive for the Philadelphia chromosome. So he had gone from 20 out of 20 to 0 out of 20. And how that came to be is the story of the Philadelphia chromosome. I first became interested in this story as a editor with a medical journal. That was around the year 2000, and I had heard these words, the Philadelphia chromosome, around. I was at a medical conference where Brian Drucker was presenting some results, and that was, at the time, that was all I knew about it. Later, when I became a writer, a science journalist, I, kind of remembered these. Sorry for the feedback. Maybe I should stand further. I remembered these words, the Philadelphia chromosome, and wanted to know, what was this, the Philadelphia chromosome? We were saying last night at a dinner with the History of Medicine folks that it would be great if everybody could name their genes with such memorable names, right? Because I could remember it years later. So I started tracking it down. I just wanted to know more. And my first question might be what your first question is, which is, why is it called the Philadelphia chromosome? That question led me to these 2 researchers in Philadelphia, Peter Noll and David Hungerford. Peter Noll was a medical doctor. He was young and ambitious and very confident, and as he told me, he was pretty sure he could kind of solve this cancer problem over the summer, that he was doing some research in a lab there, and then said, well, but that summer I got married, the Phillies won the pennant, so cancer kind of, had to be pushed to the side a little bit. But during his research, he had kind of stumbled across this method for enlarging cells. It was completely accidental, and it was because he was young and in a hurry. He had some cells he wanted to look at under a microscope, and rather than using the approved protocol for rinsing cells, he used tap water. And the tap water made the cells expand, like balloon out. So if you can imagine, I'm sure we have many people who have seen this, who don't need to imagine it, a cell in the middle of dividing, right? So you've got the chromosomes in their amazing dance, as separated as they get within the cell, and then take that cell and expand it even more because of this tap water. When he looked at those under a microscope, he could see chromosomes better than he had ever been able to see them before. And mind you, this is in the 1950s. So it's, we have to kind of take ourselves back to what did not exist today. So he wasn't really that interested in chromosomes actually at the time, because in the late 1950s, there was not this kind of cancer genes like connection going on. So he didn't pursue, what could he see now, but he thought somebody out there is bound to be interested. And there was this man, David Hungerford, who was Noll's kind of polar opposite. He was a PhD. He was not interested in curing cancer. I mean, everybody's interested in curing cancer, but he wasn't thinking he had to be the one to do it. He just loved to look through a microscope. He was just had a passion for observing and recording what he saw. And he was very into looking at chromosomes from any chromosomes he could get his hands on, he wanted to look at through the microscope. He and Noll met, I think someone introduced them, and they started working together. And they began examining all kinds of blood samples under the microscope. And one day they were looking at blood from a patient, this was at the University of Pennsylvania, who had chronic myeloid leukemia. And they noticed that one of the chromosomes in this patient's blood was a little bit smaller than normal. And again, like, to imagine what he was able to see, I mean, the arrows there show you the chromosome that he saw. It was the tiniest part of one of the tiniest chromosomes that was missing. So the fact that this man, who was one of the only people, one of very few people who could have observed this problem, was in the right place at the right time to observe it, was kind of the first of many unexpected, strange moments that eventually led to a very extraordinary treatment. So that was back in 1959, and they named it the Philadelphia Chromosome, but nothing could happen because there was no technology. I mean, other people found the Christchurch chromosome in New Zealand, and there were a couple of others that came up but kind of fell by the wayside because there was no technology to do anything with any of this research. However, obviously, things did eventually start to happen, and that was in the 1970s. So as I was researching this, somebody began to mention to me, oh, well, you need to speak with Ray Erickson, right? This man, Ray Erickson. Ray Erickson is now at Harvard. He was at the University of Colorado in the 1970s. So I called up Ray Erickson, and it's like, okay, so, tell me about the Philadelphia chromosome and your research. And he said, well, my research was all about chicken cancer. It's like, well, why were you studying chicken cancer? And he said, that's what everyone else wanted to know when I was doing this research. People used to tease him, oh, Erickson, he just wants to cure chicken cancer, which is very noble, but, you kind of think, well, maybe, turn your attention elsewhere. So I said, well, why were you studying chicken cancer? He said, well, that goes back to a man named Peyton Rous. And I'll tell you this story, which is one of my most favorite stories from this book and from all of my research I've ever done with cancer and medicine, medical drug development, because it is truly extraordinary, and especially for those interested in the history of medicine. So Peyton Rous was a scientist who went to Rockefeller University, which is in the Upper East Side of New York City. And he went there in 1909. The university was pretty new. And he was just getting his laboratory set up there when one day this woman, this farmer, arrives at his laboratory door holding a chicken. And the chicken has a tumor sticking out of its belly. And she can kind of see, this protrusion through the feathers. And she looks at Peyton Rous and she says, can you operate? It's a true story. Can you operate? Being a scientist, sure. So Rockefeller University had its first ever chicken surgery. And Peyton Rous removed the tumor from the chicken. It was a success. For a time, the chicken actually died of cancer shortly afterwards from remnants of cancer cells left in its body. But now, Peyton Rous was strangely the owner of a tumor from a chicken. And being a scientist, he did not throw it away, and instead thought, well, what can I learn from this? What can I find out? So he took this tumor and he ground it up and put it through a filter. And if you can imagine like, when you have a plant pot, that like a clay pot where the water, you can see the water coming through on the other side. I mean, it's a filter like, that's so fine that nothing can get through except, something very tiny. So Peyton Rous knew whatever was going to come through the other side of that filter was not something that was part of the chicken, right? Because even a cell, it was too thick, too big to get through that filter. So he grounds up the tumor, pushes it through the filter, and takes what comes through on the other side and injects it into another chicken. And you can probably guess what happened. That next chicken got cancer. That next chicken developed the same kind of tumor that the first one had. So Peyton Rous had discovered contagious chicken cancer, which is in itself fascinating, but not the point of the story. So now he had this contagious chicken cancer. He knew that there had been a virus, right? Because what else, right? He knew some virus had gone through into that tumor and caused the tumor in the next chicken, and this virus became known as Rous sarcoma virus, or RSV, Rous sarcoma virus. Now, what is so extraordinary about this story is, first of all, as I'm sure you can all attest to, this would never happen today, right? I mean, this farmer would be like, the paperwork alone. It's just like, I don't need the chicken, it's okay. And not only that, because that was 105 years ago now, so you can think how, just the impossibility of a farmer walking into a scientific laboratory. And also the fact that Rous sarcoma virus, the research that was subsequently done on it led to a breakthrough understanding in how cancer is caused and how to treat it. Because what happened is these two scientists, Bishop and Varmus, whose names you may be familiar with if you're in the cancer field, wanted to know, well, why is this virus causing cancer, right? Like, a virus doesn't need to cause cancer to survive. In fact, it's probably best that it doesn't, because the host will die and the virus will not get a chance to continue to exist. So, and they took out, researchers, Bishop Varmus and others, were able to create strains of the Rous sarcoma virus that didn't cause cancer. So they knew that the virus could exist without this gene, what they didn't know was a gene yet, without causing cancer, right? And a viral genome is very small, right? They don't tend to have a lot of extra stuff. I mean, a virus is like, a little squiggle of DNA or RNA and a little shell. There's not that much going on with a virus. So they're saying, well, what is this? Why is this causing cancer? How did this happen? And with the technology available at the time, they conducted an extraordinary experiment, extraordinary because it was quite labor intensive, a question that could today be answered probably in an afternoon. They had to create methods for. And so they took the Rous sarcoma virus, they took the RNA, they synthesized DNA, they made that DNA radioactive, they isolated the part of that virus that was responsible for causing the cancer, because they could make strains that did and didn't cause it, and so they knew the difference was, one portion of the genome, of the viral genome, was causing the cancer. And then they said, well, where did this come from? How did this get into the virus? So they created this radioactive probe, and the idea was they would test it against different genomes from different places and find out where it had come from. And they found out. And where it had come from gave them one of the most surprising results of their lifetime at that time, which was that it had come from the chicken. The gene that was causing cancer in the virus had actually come from the chicken itself, which is crazy if you think about it, right? I mean, this was the first time that anyone was having the notion of what we now freely refer to as an oncogene, a gene that we have naturally in our bodies that can mutate, and in that mutated state, trigger cancer. So this was an oncogene, SARC for sarcoma, had come from the Rous sarcoma virus. So that was the first revelation that came from Peyton Rous's chicken. And then Ray Erickson said, well, what kind of gene is it, right? He wanted to know, okay, you have this oncogene, what does it do? What does it do normally? And what he found out was that this gene encoded what's called a kinase, which is a type of protein. And kinases, we have many of them in our bodies, and they are, they trigger pathways to start in our body. They give a signal for a process to start and a process to stop. Now, what does all this have to do with the Philadelphia chromosome? Well, of course, in the meantime, also in the 1970s, people are finally having breakthroughs, finding out more about why this chromosome was too short. What happened? Where did it go? And Janet Rowley at the University of Chicago was the one to finally figure out what was going on, which was that two little bits from two chromosomes had swapped places. It was a translocation. And her story is, I mean, if you ever are just, next time you're procrastinating, go look up Janet Rowley on your computer. She's a fascinating person. And she used to sit cutting up. She had pictures of chromosomes that she would cut up and lay them out on her dining room table, and her children were like, why are you playing with paper dolls? And so she found out it was a translocation, and people didn't actually believe her at first until she found another translocation and another translocation. And these two genes, the swapping that went on involved two genes, one called BCR and one called ABL. No, that's wrong. Sorry. But in the swapping, one of the genes, ABL, moves from one chromosome to another, bringing it next to BCR. Okay, so that was the translocation, was ABL and BCR coming next to each other. And ABL is a kinase, right? So it stops and starts. It gives the signal for things to stop and start. And ABL and BCR, two genes, totally normal in our bodies, and like two children who are all sweet and well-behaved when they're playing separately and come together and get up to all kinds of trouble. BCR and ABL are like this. When they come next to each other, ABL goes haywire. ABL is the kinase, and it goes haywire. And instead of ABL is a kinase that triggers white blood cell production. And what happens when it comes next to BCR is that it never shuts off, right? So you heard about Gary Eichner, who had way too many white blood cells in his body. It never shuts off because the signal just doesn't stop. So in the translocation, ABL mutates, it becomes an oncogene, and it just doesn't stop, and people end up with chronic myeloid leukemia. So you see, this chicken, the legacy of Peyton Rous, the Rous sarcoma virus, leading to not only the discovery of oncogenes, but also uncovering the role of kinases. But the thing is the story, of course, doesn't end there. And this was, kind of the part of the magic of researching this was, okay, but then what? And it just keeps going. It keeps unfolding. Because in the mid-1980s, when this kind of riddle of the Philadelphia chromosome was solved and people finally understood The translocation leads to BCR-ABL, which leads to white blood cell production overdrive, which leads to CML. There were some people, Brian Drucker was one of them, who started saying, well, can we make a drug to target that, right? This was the first time, like, the mechanism was known, right? There was a concrete sequence. This is how the cancer is caused. And up until then, cancer treatment was, well, let's try this, figure something out to make it better. Now there was the root of it fully described, and some people saying, well, we know what's going on, we know the mechanism. Can we create a chemical that will kind of throw a wrench into the clockwork? And so Brian Drucker was one of the people asking this, and he happened to meet some other people who were also asking this question, who were working in the pharmaceutical industry. And at Ciba-Geigy, which would later merge with Sanofi and become Novartis. So at Ciba-Geigy, they did this. And I want, again, another kind of fascinating whole storyline there. They actually made a chemical that when a person swallows it, it can block this mutant kinase from working. So the healthy cells are able to continue normal white blood cell production, and those housing the Philadelphia chromosome are unable to continue proliferating. So they did this. It was in Switzerland. They made this chemical, and they sent a bunch to OHSU, to Brian Drucker, who was by then at OHSU. The timings worked out that he arrived here right when the chemicals were ready. And they sent him a few to test. They sent him 4. One of them was the one, and then they sent him a few other ones. One was a complete fake. And he saw that one of these compounds was it was killing cells in a petri dish in his laboratory. And now seeing that would be, in a normal circumstance, a great impetus to move something on, right, to an animal trial, right, hopefully to a clinical trial if it works out. But things moved very, very slowly, and Brian Drucker had to keep saying, okay, like, when is, this compound is doing something, like, let's move it along, guys. But they didn't. Eventually, animal studies got underway, and those two ended up taking way longer than they normally would. And hearing about this from Brian when I came to interview him, I wanted to understand why. What happened? And it actually took 18 years from the time that compound was synthesized to the time that this drug, Gleevec, was available on the market was 18 years, which is a very long time in drug development years. So the animal studies did start, and the first news to kind of come back was that there were some side effects, right? There were some risks to it. There were some dogs who developed liver problems. There were some blood clots, and they wanted to stop the trial at the pharmaceutical company. There was actually, so there was a man at Novartis named Alex Matter who was fighting for this drug, while there were other people at the pharmaceutical company saying, no, we can't, this drug isn't safe. He told me there was actually a man from the regulatory division who told him, not over my dead body will this drug ever be given to people. And Brian Drucker is hearing about this liver toxicity and that they want to stop the trial, and he's saying, you're crazy. You have a drug that could potentially save lives. You have a drug that could be effective. you have to give it a chance, right? We're doctors. If somebody comes in to our clinic in a trial and develops liver problems, we're not going to keep giving them the drug, right? Which is what they do in an animal study. They were giving the dogs extraordinarily high doses of the drug. And a clinical trial starts out at, minuscule amounts of it. You have to give it a chance. You have to try this. You have an ethical imperative. But of course, the problem was not just the fear of liver toxicity or other issues. There was also a financial concern, right? So CML is extrememly rare. Not the rarest of cancers, but it's rare. It's diagnosed in up to 70,000 people worldwide each year. So to put that in proportion, there's about 250,000 people diagnosed with breast cancer in the United States each year, right? So like breast cancer, CML. So the pharmaceutical company is making their projections and saying, we're not going to recoup on our investment, right? They're using a model of chemotherapy and how cancer treatment tends to go to make their calculations, and there's no way they're going to recoup on the investment. Finally, of course, the phase 1 trial does begin. It began in 3 locations, here and at UCLA and at MD Anderson Cancer Center. And we are very fortunate to have in the audience today Judy Oram, who was one of the very first patients to take the drug here at OHSU. She's here if you want to, I don't know if you feel comfortable, but so the phase 1 clinical trial began. There were about 30 patients altogether, and the results were unlike anything any of them had ever seen. In nearly every single patient, the leukemia was going away. The response rate was 100%. And this is absolutely unheard of when it comes to cancer. And the side effects were minimal for most people. And they reported these findings at a big medical conference, right? It was a huge moment. But then still, even after that, it took a long time, right? The phase 2 trial, so it goes from phase 1 to phase 2. Sorry if I'm telling you all information you already know, but it goes from phase 1 to phase 2 to a larger study. And it was still just, like molasses. Brian Drucker had a waiting list of 100 patients long, and every time he was given permission to enroll another person onto the clinical trial, he had to choose the sickest patient to enroll. And in the meantime, they're trying the drug out, not just on newly diagnosed patients, but on advanced patients. And people are being wheeled in here. People are being pushed in on wheelchairs. One woman who had selected her burial plot and was, preparing herself to go, her husband had begged her, can you just please try this one last drug? And she's alive today. She was out of her wheelchair within weeks of starting on this drug. But still, even with that, the phase 2 trial is just not ramping up. And Brian Drucker at one point got an email from a woman named Susan Mcnamara, who was in Canada. Susan Mcnamara was very sick. She was basically existing between her bed and her computer, a couple feet away. And she had lost tons of weight. She was taking at the time the standard treatment for CML, which was interferon, which is basically, as far as side effects go, it's like giving yourself the flu. I mean, people talk about their year of sleep, essentially taking this drug. It can also cause depression. It's a very difficult medication. Now, another incredible moment in the history of this drug was the fact that this development, this moment in time when this phase 2 and phase 1, for that matter, trial was going on, correlated exactly with the rise of internet chat rooms. So you have people who are, for the very first time, if you can take ourselves way back to the time when we weren't sharing everything online, this was the beginning of that. And so these people in CML, right, it's a rare disease, right? So if you want to meet people who have the same cancer as you, it's unlikely to happen like in your hometown, right? You're not meeting for coffee to talk about side effects. But on the computer, right, in online forums, well, that could bring people across the world into contact with each other. And so people start doing that, and they're talking about their side effects, and how do you deal with this, and how do you deal with that, and what's your this count and your that count. And then there are some people on these chat rooms saying, I feel great, right? I'm on this clinical trial, and it's, you know, my blood counts are down, I have no side effects. And, you know, there's all these people like, what? You know, and people whose doctors didn't know about the drug, right? So patients are coming in. This was the beginning of, you know, educated patients coming in and saying, I want this drug, right, with their printout of discussions online. And, you know, some people being told, oh, let the other people test it out first. You know, it was just a whole other era, even though it was not too long ago. And so Susan Mcnamara is reading these online forums, hearing about this drug, and she calls up Brian Drucker, or emails him, and says, you know, can I come? Can I get on the trial? And Brian Drucker writes her back saying, you know, I'm sorry. I don't have the drug to give you, and I don't know when I will, or if I will. He was at that time, despite the phase 1 100% response rate results, still wondering if the company was ever going to even make the drug. And Susan Mcnamara, you know, so Brian Drucker, when he wrote her back, he said to her, you know, he was out of ideas, and he said, well, maybe there's something you can do. Right? And she said to me, like, how, you know, this response was like, sent this kind of shaft of light down to her. You know, she was going to die. And she just said, there is no way that there exists in this world a drug that could help me that I'm not going to get. So she started an online petition, another, you know, new thing at that time, and had, you know, hundreds, thousands of signatures petitioning the drug company to expand the clinical trial, basically. And having received this petition, for whatever that might have meant to the drug company, the clinical trial was soon opened, and Susan Mcnamara came out to Portland, enrolled on the trial, spent a few weeks out here, like eating lots of food and gaining lots of weight and getting better. And she actually later, she's also still alive, and she actually ended up returning to college and becoming a scientist after that. So these are a few of the moments that eventually led to the approval of Gleevec, which now has been on, you know, it has 10-year survival rates and has held up over time. It is one of the most successful drugs, if not the most successful drug ever created for the treatment of cancer. And really, you know, what I grew to love about this story, about this moment in history, is, you know, how firstly it pulls the curtain back on drug development, on medicine, on so many questions that we all have about why things are the way they are, when it comes to healthcare. And just even more than that, for me, just shows the miracle, not just the miracle drug, really, but the miracle lengths that people are willing to go to see something they believe in get a shot in this world. Thus the book. So I'll leave it there as far as not giving too many stories away about the book, but I'd be really happy to answer any questions that you might have about any aspect of what I've said or other parts of the story. That's okay, if we still have time. Thank you. Thank you.
We're asking people who have questions to please come up to the mic. This is being video recorded, and we want to make sure that we get your great question as well as Jessica's answer.
Hey, Jessica. One of the things I really liked that you made a choice in your book was to tell all the scientific backstory before Brian and humanizing his role and not making him the hero character of everything and the man who discovered everything. Was that a difficult choice to make? Because it could have been easier to just put Brian on a pedestal and focus it all around him and not the complex backstory of dozens of scientists that you did go through.
Yeah, thanks. That's a really great question. So I'll say a couple things in response, which is I was actually very determined to include all of that backstory, even though it gets very technical, and I was quite struggling, at many points, like, okay, I think I understand it. Oh, wait, no, I don't. Wait, no, I've got it now. And, there's a lot of complex stuff in there. But there were a couple reasons why I wanted to make sure to include that. One was because I really wanted anyone reading the book to, I felt like the payoff would be better when we get to the point of meeting Judy and hearing what happens with her and other patients along the way, that having the scope of history behind those moments would just make it that much richer of a reading experience. It was also difficult sometimes to